What I'd now like to talk about is the broader strategies of how do we expand the number of projects we're working on from an internal perspective and an external partnering perspective. So I'm going to talk about, firstly, the sweet spot that we've identified for our internal development projects. These will encompass G-protein coupled receptors with well understood but complex pharmacology, and they will be implicated in fibrosis, inflammation or oncology. So a really well-defined sweet spot that leverages not just the capabilities of our iBody technology, but also our other networks of internal skills and external subject matter experts. I'll then talk about the processes we're going to use to develop platform partnerships outside of that sweet spot, but in areas that continue to leverage the unique capabilities of the iBodies, and our partnering strategy for AD214, but it will be the same process that we're going to use for any of the other internal development candidates. So the starting point for our internal pipeline is a rigorous and robust product selection process. I'm not going to go through this in detail. Suffice to say that there are 5 stage-gated steps, and each of those represents an increasing level of rigour so that when we commence an internal development program, the board have signed off on the investment from the perspective that we know where this is going, we know what the target product is, and what the milestones along the way are, so we can continue to decide whether that warrants continued investment. Right now we're in the rapid screening and desktop validation phase. And so you can see we have a little bit of time to go before we're ready to announce specific targets, but we are actively working on screening our next set of internal pipeline projects. The definition of the sweet spot. Essentially we have decided that our internal sweet spot will be difficult to drug G-protein coupled receptors. They must be clinically validated. So this means there must be a very, very clear linkage between engaging this receptor and modulating disease. The indications will be in fibrosis, inflammation and oncology, and these are some of the largest unmet disease categories in the world today. And we must have evidence that the therapeutic area and the receptors specifically are not being adequately addressed with small molecule or antibody therapeutics. In other words, we actually want to see that other people have tried to drug these targets and failed. And more importantly, that we understand the reasons for failure. So all of that means that we're hitting difficult to drug targets, but we're taking certain steps to de-risk our product selection and leverage our own internal capabilities. So the type of early screen process you can see is the matrix on the right-hand side. Now, there are a whole bunch of other really interesting and cool targets out there. We've had a long debate around ion channels, for example, which are even more poorly drugged than GPCRs. For the moment, our aspirations are to partner to access that particular target class, and we'll find partners who already have expertise and interest in those particular receptor classes. That's not to say they won't come into our internal pipeline down the track, but for the moment our sweet spot is GPCRs. And what are they and why are they interesting? GPCRs are one of the most diverse group of membrane proteins in humans. They're kind of like an inbox to communicate messages from the outside environment into a cell. And there's all kinds of different messages. It's a bit like your inbox. There's everything from spam to the most important financial information or what your loved ones and family are doing at school, right? So these GPCRs have to filter all of those messages. And when they find the right one or ones, because often they are capable of processing multiple of these messages, they act as a switch, turning on or off internal cellular processes. And so that's how they work. So you can start to appreciate that these are going to be really complex beasts. They're going to take signals from multiple different sources, and many of them have multiple different ligands or binding partners in nature. So they respond to half a dozen potential signals. They will also trigger multiple different pathways, biochemical pathways as a result, and they might be very similar to another GPCR that might be even on the same cell that triggers a whole separate different cascade. Now typically only one of those pathways will be involved in disease, so the importance of being able to hit these things really specifically and really selectively is where our iBody platform is particularly advantaged. And we've seen that with the way, as I mentioned earlier, with the way we've been able to get very selective pharmacology with our CXCR4 i-body, but we've also seen that with several of the other i-bodies we've worked on in the past. These GPCRs represent 12% of all known drug targets, but more importantly, there's almost a third of approved drugs today target these receptors. So that gives you an idea of just how important they are to pharmacology today. The challenge is that many of those drugs haven't proved to be particularly effective. So just to highlight this story, there are about 400 GPCRs, known GPCRs, other than, there's another 400 that are involved in your sense of smell, by the way, but 400 that are potential therapeutic targets. Just over a quarter of those have been drugged today, which means there's 3/4 that don't have approved drugs against them. There's another maybe 20% where there are drugs in the R&D pipeline, and there's over half of them that don't have drugs against them. And in some cases, we don't even know how these work. So they'll drop out of our filter because the pharmacology is not well enough understood. But the point being is that, a recent review article described these as there's over 200 GPCRs with broad, untapped therapeutic potential. And of the ones that have been drugged, plerixafor is a good example, the only one that's targeting CXCR4 today, and we know that it doesn't do everything that we need it to do to deal with CXCR4 modulated disease. So this is the opportunity we've got in front of us. And it's feasible to build an entire GPCR-focused portfolio around the indications that we've talked about. So these are all of some of the GPCRs that are implicated in fibrotic disease. And I'm not going to go through in each one, simply to be able to say that there's actually a number of them. Fibrosis is a complicated disease, and it's a bit like cancer, and there's going to be room for multiple different approaches to target this disease. Many of them are GPCRs, and so by focusing on this disease area, we not only leverage our i-body capability, but we also leverage everything we're learning about fibrosis. We're also leveraging the same clinical experts. We're also leveraging the same preclinical experts that are investigating these types of fibrotic mechanisms. So I hope that gives you a little bit of a sense to say this focus on GPCRs makes sense. There's sufficiently wide white space out there for us, and this focus on these therapeutic areas is not constraining our opportunities significantly, but providing a degree of focus that allows us to build one program on top of the next with synergies between them. Now, over the 12 years of the company's existence, we've found over 20 different complex targets to which we've been able to generate i-body binders. And just about half of those have been GPCRs. You can see on the right is where these are up to in our discovery process. To get from discovery idea to proof of concept is anywhere between 12 and 24 months, depending on the complexity of the target and various other factors. So by starting with the binders that we've already identified, we can save 3 to 5 months. So that's why we're starting there, because we know there are binders in our libraries, and we can save a little bit of time if any of these pass our rigorous product selection filters. And so what we're doing at the moment is taking those existing targets, plus a few new ones that we're working on. We've engaged one of the folks who did this work for Ablinks years ago, and for Arginex more recently, is helping us with this target selection process. They go into the funnel, they go through that rapid screen that we talked about. Anything that drops out, particularly if it drops out because it's not a GPCR, because it's not in our target indication space, or because it doesn't require the particularly exquisite selection of pharmacology, goes into the potential partnering opportunities. We don't write it off altogether, we'll just work with partners to work on those ones. The aim at the end of the day is we want to select 5 that get through that 5-stage process that we can then justify putting into internal development. We have 9 of our existing potential targets are still under active evaluation. And supporting all of that is some work that will be going on in the lab processes, and that Dallas will talk about with our i-body 2.0 program to make that discovery process shorter and more efficient. So that's how we'll get to the selection of targets. While we can't announce specific targets today, I hope that gives you confidence that we have a pretty robust process and a very clear focus about where we're going to go looking internally to leverage our skills. This is what our pipeline hopefully should look like in 2 to 3 years' time. So the solid brown bars show us where we're up to today. Essentially, we've got AD214 heading into phase 1. We've got discovery work going on in the second and third indications, and we're working on discovery with the GE collaboration. And everything else is exploratory. So we're hoping to be able to extend that and have multiple programs in the clinic, multiple indications into IND-enabling studies with AD214, and multiple new partnerships. So in 2 to 3 years' time, we should have a much more robust product pipeline. Once we have programs in internal development and for targets outside of our sweet spot, we need to be able to monetize those, which means we need to be able to enter partnerships. So how do we go about partnering? These are processes. They don't just happen. And they're actually 2 different processes. So the top of this slide shows the process for partnering our platform. These are co-development deals with other companies who are bringing a target that want to work with us on discovery. Typically they'll be like the GE deal, a small upfront for access to the pipeline, access to the platform, I should say, and then research fees along the way. To get from prospect to contract is, I estimate, roughly a 6 to 12 month process, and our objective is to have a continuous stream of prospects filtering through this pipeline. This is pretty much like your standard selling process for any business or any organisation, and our platform is a service in that sense. So my internal metric is how many projects have we got at each stage of this pipeline and how quickly are we moving them through the pipeline. Relatively standard and pretty straightforward. The product licensing process is slightly different. Here our objective isn't to move people progressively through the pipeline at different stages. Here what we want to do is have a number of people through the pipeline to a point so that we can create an auction when we get to a partnering window. And the partnering windows are typically going to be the end of phase 1, the end of phase 2. And at that point we want 2 or 3 different companies at least interested so that we actually create competitive tension to get the best possible licensing deal. And so for that reason the process is slightly different, and the metrics are less around how fast we're moving people through, but are we actually building critical mass of potential discussions for those key timeline points. And investing business development time and resources is a priority for us at the moment to fill these pipelines. We continue to be confident in the potential of IPF and for being able to partner AD214. We've talked about the incidence of disease. We've talked about the fact the market is a $3 billion market today with only 2 drugs and they don't work particularly well. And we know that deals are still happening. There have been 3 big deals in IPF just in the last 9 months. So that was a very quick cooked tour of how do we build out the pipeline and monetize the assets that we're building. And with that, I will now hand over to the best for last, Dallas.